So what I'm going to do right now is I'm going to help you successfully write the Wisconsin Fast Plant Lab. And although we took all different types of data, our focus here is to understand the phenotypes that were produced when the F1 generation plants that we planted were crossed with themselves. So let's step back a little bit, knowing what we have done in terms of collection of data for these objectives will help us figure out what we need to do. So what we did is we started with seeds that we've got from the company, the Wisconsin Fast Plant Company, and we planted them. Now they were offspring of the parental cross here. And you can see that we are homozygous dominant here for the purple stem, and we are homozygous for the green cotyledon, which is the little leaf, and we are homozygous recessive, so non-purple right here, and of course we are a yellow leaf for the cotyledon. So purple, green, non-purple, and yellow. So those are our phenotypes. Now we crossed them, we didn't cross them, but the company did. They produced seed pods. They opened the seed pods and they gave us the F1 offspring. We planted those. And when we did so, we noticed that all of our plants were purple stems and green leaves. Okay? And what we did was we crossed them randomly. And what we got was 4 different phenotypes. And you can look and see what those are. Okay? We've got, let's say, taking A and L, we got A and L. And we could have gotten what? A and L, big A and L. We could have gotten big YGR and another big YGR. And that would have been what? That would have been purple stems, and that would have been green leaves. Okay, what else could we got? We could have gotten big A and L, little A and L, and then YGR. And we can keep doing this, okay, to get all of our different phenotypes. I'm not going to do that, but this would also give me purple and green leaves. And you can keep mixing and matching to do this. In fact, a better way to do this would set yourself up with a Punnett square. Okay, now how would you do that? Well, remember, we have parents. In this case, we have these. What are the possible 4 combinations of alleles that we have? Well, we have, if we were to separate this, okay, we can have big A and L and capital YGR. What other gametes can we have? Well, we can have big A and L and little YGR. All right, these are going to go together. These are the gametes. You're picking one out of the 4 combinations, assuming there is a law of segregation and independent assortment here, because these are 2 different alleles. Okay, what else? Well, I can have little A and L and big YGR. And that's another combination of gametes. Okay, and then I can have what? Little A and L and little YGR. So all I did was take this paternal or maternal plant, and I broke up the 4 alleles into the 4 combinations of gametes. And sometimes you can think of this as the dad, the sperm, or the eggs. And I'm going to do the same thing. Well, because the other parent has the same exact genotype, it's the same combination. A and L, YGR, A and L, little YGR, A and L. This time that'd be small, so I have to make sure I do that correctly. Sometimes I should use the lowercase, but I'm tall. A and L, big YGR. and then A and L, little guy, and then little y, g, r. And I probably should fix the ones over here, not to confuse anybody, although I'm good at confusing myself. All right, in any case, A, and do that again. Okay, so what I did was I wrote down all the 4 possible combinations of gametes. These could be the eggs if it's the female. And what we're going to do, or what you're going to do, because I'm not going to finish this, you can put these together. And how would you put these together? Well, there's going to be 16 combinations, right? 4 columns and 4 rows. I can do one for you. Big A and L, big A and L. And then big YGR and big YGR. And you can keep going down, and you're going to get 16 different combinations. And knowing that the purple is dominant, this is purple, and that the YGR, the green, is dominant, you can figure out all of the different, and there'll be 4 different phenotypes overall. There'll be several, more so, more than 4 different genotypes, and as you know, you can have different genotypes with the same phenotype. Okay, case in point, if I was to do the second part of this column here, big A and L, all right, and with another big A and L, now I get YGR and little YGR. Well, as you can see, these are different genotypes. This is homozygous for the purple stem, but this one's heterozygous for the leaf, where this is homozygous. However, because the YGR capital, or green, is dominant, these are both purple and green. So you're going to have to make sure that you count correctly. All right, and you're going to come up with some ratios. And obviously, I'm not really hiding anything. You're going to come up with a 9:3 to 3:1 ratio of the 4 different phenotype combinations. Okay, now what do you do with this? Okay, well, now that you've established this in your data, what you're going to do is you're going to go to your observations. So somewhere, okay, in your lab, you have 4 different places of observations. So let's think of all the possibilities. We can have, let's just start here, and we can have purple stem and green leaves. That's what we have here. We can have purple stem, and we can have yellow leaves. We can have green leaf, that means non-purple, and then, I'm sorry, green stem, which is really no color, and then green leaf. Then we can have, again, non-purple, which we'll just write green. And if you don't like that, I'm not trying to confuse anyone, so let's make that non-purple. So non-purple and green stem, and then non-purple, yellow leaf. Okay, so these are the 4. And you may have some numbers from your total class data. I'm going to put in some different numbers here. Let's say I've got, I don't know, 70, 30, 10, and 5. Okay, so that was total. That's the total that I got for my class. I'm making this up because some people don't know what to do with this, because at some point what you're going to have to do is you're going to have to see if the actual observed that you got from your class data by people counting the frequencies of the different alleles is the same as the expected. Now, this is very important. This Punnett square that I did not finish, that you should, okay, is going to give me these 9:3 to 3:1 ratios for these particular phenotypes, not in any particular order. So you got to figure that out. You got to figure this out from the Punnett square. Now, what's really important here is to note that we would get a 9:3 to 3:1 exactly if the Punnett square and our test and our alleles on the Wisconsin fast plants are in fact on different chromosomes. See, Mendelian genetics is the idea that these traits, this could be what? Big A and L, this could be little a and L, right? In meiosis, they duplicate. Okay, it's the same one, A and L, little a and L, and then they get pulled apart so that in anaphase, these get pulled away. And it's just by chance to know if you're going to get the A and L gamete or the big A and L gamete or the little a and L. Total chance how these align. And that's why we call this the law of segregation. Now, when you think of another allele, okay, let's make another tetrad. And let's make this YGR, big YGR. Now, little YGR, little YGR. Total, what 4 combinations can you make? It's important you realize that these also can align. Couldn't this be on that side and go with that cell? Couldn't this be on this side? So you can see that the law of segregation and the law of independent assortment, meaning that these alleles, if on different chromosomes, will give these ratios because it's all about chance to see which one aligns up on the right side or left side. So segregating is total chance, and the law of independent assortment means we're on different chromosomes. Okay, so all of that we're doing here is going to match these ratios if we follow Mendelian genetics, which is based upon, luckily for Gregory Mendel, of pea plants or alleles that are not linked. Okay, so your null hypothesis is very simple. Very simple. And what you're going to say every time you make a null is that your expected will match the observed. We're going to pick this ratio as the expected because in my null hypothesis, I'm testing that my outcome of my second planting of my generation of Wisconsin fast plants, all right, are in fact on different chromosomes. That's really what I'm testing in my null. And all I have to say is that the expected frequency of the 4 phenotypes will be in a ratio of 9 to 3 to 3 to 1. That's it. Always make your null hypothesis with the expected frequencies that you want to pick. We can make the null anything we like, but we're going to test the idea that the 2 different alleles on our Wisconsin fast plants, are they really on the same chromosome? So we're going to make the null state that they are. Okay, because we're going to make it expected. That's what we do with the null. We choose an expected outcome or expected frequency, and then we test to see if it's right. So that's what you're doing here. My expected phenotypes are 9 for one of these 4, 3 for another, 3 and 1. Okay, and that's what your null hypothesis. And then we're going to run a chi-squared to do what? To evaluate the strength of this null. See, what I would say is very simple. My hypothesis is that I expect the purple-green to have a ratio be in a certain frequency. And now, can I say a 9 to 3 to 3, 1 frequency? Sure. I could say purple-green will be 3, the purple-yellow might be the 9, who knows? You have to figure that out in your Punnett square. So it's understanding that the null is about a certain expected value that you are defining. We're going to choose this as my expected, so I'm going to test my outcome with this expected. And the chi-square that you get will do what? Give me that strength of the argument that these are a 9 to 3 to 3, 1. Okay? Now, some people having some other issues say, well, how do I do this exactly? Okay, well, here's how you would do it. Take your observed, and let's go with the observed frequencies that we actually got from looking at the phenotypes of the plants in the second generation. Okay, we know the expected is 9 to 3 to 3, 1. Let's pretend that this is supposedly supposed to be 9, this is 3, this is 3, this is 1. I don't know, you have to check. How do I do this? Well, your AP biology equation sheet shows you that your null hypothesis is what? Well, or I should say your chi-squared is what? Chi-squared is equal to the sum of all the individual observed minus expected squared over the expected. So we're going to do this 4 times, right? We're going to add these sums together. How do I do this? How do I get my chi-squared? How do I get my values? Well, I'm going to take my observed and minus it from my expected. And I'm going to do that for every single frequency. So my observed is 70. And I minus it from the expected. Now, what's the expected? Well, if in fact this is supposed to give me a 9 to 3 to 3, 1 ratio for purple, green, and I'm not sure if it does, you have to check, okay, I've got to find an expected value for that. Well, the first thing I'm going to do is add up all of my plants. 70 plus 30 is 100, plus 10 plus 5 is 115. So there's a total of 115 plants that were observed. Okay, now I need to find the number out of 15 that'll give me a 9 to 3 to 3 to 1 ratio. Now, forget about a 9 to 3 to 3 to 1 ratio. What does that mean? If it's a 9 to 3 to 3 to 1 ratio, isn't that really 9/16? You say, well, how did you get that? Well, 9 of the phenotypes in your Punnett square up here, 9 of your choices or genotypes would give you that purple-green phenotype. Or just count these numbers up from your ratio and you get 16. So 9 out of 16 would do that. Okay, so 9 out of 16. Let's go find that number. So 9 divided by 16. 9 divided by 16. And I get, let's say, 0.56. I'm going to round. So I'm going to take this 115 number, that's a total number of plants, I'm going to times it by 0.56. Now what's that going to give me? Well, it gives me 50%, 56% of 115. So 0.56 times 115, and that gets me 64.4. So 9/16 out of the entire 115 is 64.4. You see, I had to calculate the expected from this ratio. Do not put 9 here. That is not correct. Okay, so let's walk it through the next one. 30, and of course what we do is 70 minus 64.4. Okay, we're going to find that number. We're going to square it. Okay, we're going to divide by the expected, which is 64.4. All right, we're going to find that entire number, and then we're going to add it. See, it's the summation of the other 3. So what's the other 3? Now I'm not going to do all 3, but let's do the next one. So we got 30, which is the observed, minus the expected. Well, if this phenotype in your Punnett square was 3 out of 16, or it was a 9 to 3 to 1 ratio, it's 3 out of 16 for the same reasoning. Let's go find that number. 3 divided by 16 is equal to 0.19. So I take 115 and I times it by 0.19. And what does that give me? What's 19% of 115? So 115 times 0.19. And I get 22%. Let's just round off for the sake of ease, 22%. And that's my expected divided by the expected, 22. And you're going to square that. And I'm going to do that again for 2 more times. So I'm going to add it to 2 more times. And when I add that entire thing up, that's my chi-squared. And then I'm going to put it into my critical values table and see if my p-value is equal to or less than 0.05. And if it is, that means that the probability is too low to support the idea of the null hypothesis, which meant that these observed are really these ratios, and I'd have to reject. Now, if my probability is greater than 0.05 based on the chi-squared value and the critical values table that I look at the AP equations, then I can say that there's a good chance that our null hypothesis is true and that these ratios are indicative. And that's what you're doing in the lab. If you come up to reject the null hypothesis, you need to discuss what other things besides chance, flipping a coin, has made your numbers different. And of course, that's what we talked about all last week with our forms I just gave back. The idea that genes sometimes may not be on different chromosomes, and that crossover is happening. Okay, so think about the idea of linked genes and explain the possibility of how these are interacting. Hope that really helped.